Good afternoon, everyone. Welcome to our Thursday afternoon webinar. I'm Melinda Bachini. I'm the Director of Patient Services with the Cholangiocarcinoma Foundation. I'm also a 13-year survivor of stage 4 intrahepatic cholangiocarcinoma. And I couldn't be more excited today than to be able to introduce you to Dr. Eric Tran, who received his PhD in biochemistry and immunology from the University of Victoria in Canada, where he studied the mechanism of how a protein called interleukin-2 caused T cells to grow. Eric then trained at the NCI with Dr. Stephen A. Rosenberg, a pioneer and world leader in cancer immunotherapy. With Dr. Rosenberg, Eric developed the first adoptive T cell therapy that targeted cancer mutations and demonstrated that this strategy could lead to clinical responses in some patients with metastatic cancer. Eric is currently an associate member in the Earl A. Childs Research Institute at Providence Cancer Institute in Portland, Oregon, where his team is trying to better understand why adoptive T cell therapy works in some patients, but not others. His team is also focused on developing more effective T cell-based therapies for patients with solid cancers. And on a personal note, Eric is the research scientist who studied my T cells, and I am only here 13 years later because of this man, and I'm so very grateful and excited that you're here, Eric, and I will turn it over to you. And just to let everyone know, we will open it up for question and answers at the end of the presentation, so feel free to put your questions in the chat. Thank you.
Great. Thank you, Melinda, for that kind introduction. And it's always wonderful to see you. And thank you for the invitation to present today. And so today I'd like to tell you a little bit about some of our work on T cells and mutations and how we're harnessing T cells to target mutations. And I'll be kind of telling this story through the lens of, Melinda's story, actually, in terms of what we did in terms of generating her treatment. And some of the data that has not been published yet. And so I do have some disclosures. I do consult, and I'm on the advisory board of Turnstone Biologics. These are all cell therapy companies. And so just to begin, so what exactly is a T cell? So a T cell is a type of white blood cell within our bodies that protects us from microscopic pathogens such as viruses and bacteria. And so if we just kind of do a simple schematic here, here's a T cell on the left-hand side here. And so within each one of us, we have billions upon billions of T cells that are circulating within our bodies and embedded in our tissues. And there are many different types or subtypes of T cells, but one of the defining factors of a T cell is highlighted here. So each T cell has something called a T cell receptor that's embedded on the cell surface. And within each one of us, there are about 1 to 20 million different T cell receptors in our T cells. And so each given T cell has one unique T cell receptor. And the reason this is important is that each T cell receptor can actually recognize a unique pathogen, that virus, or other foreign invader. So just for an example, so if our bodies are infected by a virus, let's say this is a flu shown here, infects cells in our body, those viruses gain entry into a cell. But within that cell, the virus can actually be processed, chopped up. The proteins of the viruses can be chopped up. And these peptides, we call them, can actually be presented onto a molecule on the cell surface. In this way, a cell that is infected can flag itself as being infected because now it's presenting this virus peptide. And within the billions of T cells that are circulating within our body, there's going to be a small subset of these T cells that have the right T cell receptor shown here that can engage, that can recognize this specific viral peptide. And this, we call it an MHC molecule. And upon recognition, this T cell receptor can trigger this T cell to kill the infected cell. And so this is an example of how a T cell can, recognize a virally infected cell. But why are we interested in that? How is that relevant? Well, it turns out that some T cells in our body can recognize cancer cells. And so again, within the billions of T cells within a patient, a small number of those T cells will have the right T cell receptor that can target cancer cells. And so this brings me to the type of immunotherapy that my lab is exploring, and it's called adoptive cell therapy, or ACT. And so this is a type of immunotherapy. Immunotherapy is essentially a therapy that tries to harness the body's immune system, where immune cells, typically T cells, are removed from a patient, grown and manipulated in the lab, and then reintroduced back into the patient. And the concept behind this approach is actually quite simple. It's that if you can infuse a large number of these tumor reactive T cells, hopefully these T cells can then overwhelm and eradicate the tumor cells. Currently, there are two major types of adoptive cell therapy. One uses something called tumor infiltrating lymphocytes, or TIL for short. And the other uses T cells that are derived from patient's blood. But what happens is that those T cells are engineered, genetically modified to express receptors that can target the tumor cells. And so in that way, we're redirecting the T cells from the blood to now attack the cancer cells. So currently, those are the two major types of adoptive cell therapy. And so historically, the first generation approach of adoptive cell therapy in the setting of solid cancers was pioneered by my former mentor, Dr. Rosenberg at the NCI. And so this approach used tumor infiltrating lymphocytes. And so in this approach, a patient with cancer comes into the clinic. The tumor is resected, surgically resected. That tumor is then taken into the lab, where it's minced up into small fragments, typically 1 or 2 millimeters in size. And those fragments are then cultured in the presence of something called interleukin-2, IL-2 for short. And IL-2 is a T cell growth factor. It's actually one of the most potent T cell growth factors. And so in the presence of interleukin-2, the T cells within the tumors will grow and divide, and they'll expand. And then select cultures that we think are the most effective can be selected and then further expanded from millions at this stage to billions. And these billions of T cells then can be reinfused back into the patient, again, with the expectation that a large number of T cells can overwhelm the cancer cell. This approach is actually quite elegant in its simplicity, right? So the idea is that some of the T cells are within the tumor, are actually recognizing the cancer cells, but they're for some reason being inhibited and they're losing the battle. So if you can take these T cells out of that environment and then grow them up to large numbers, then again, this army of T cells can hopefully eradicate the disease. So this TIL therapy, adoptive cell therapy, was really first studied in the setting of metastatic melanoma, which is the deadliest form of skin cancer. And I just want to highlight, the main findings from decades of work. Is one of the main lessons that we learned from TIL therapy was that TIL can be curative. So when patients with metastatic disease, so meaning disease that it's not just to the skin, but has metastasized to the liver, lung, and sometimes even brain, when you use this TIL therapy, as I've described, about 50% of these patients can experience tumor shrinkage. And of those 50%, a subset of those will actually go on to be effectively cured. And when I say cured, there are some patients that are out 20 plus years that have no evidence of disease. So about 20 to 25% of these patients will experience these long-lasting, durable responses. So that's really lesson number one. Lesson number two is that these TIL, or these tumor-infiltrating lymphocytes, they're also known as, can recognize mutations, and these mutations are also known as mutated antigens or neoantigens. We think these TIL that recognize these mutations likely played a major role in the clinical responses. And the reason why we think that is because, first, when we studied the T cell products that we gave to these melanoma patients, we could find T cells that could recognize the patient's own mutations. And so there's a strong correlation that, well, we infused T cells that recognize mutations and the patient's cancers shrank and disappeared. So it seems likely that these mutation reactive T cells were responsible for the clinical activity. And so what does it actually mean when I say a T cell recognizing a mutation, and what exactly is a mutation? So if we just look at this cartoon here, the reason why cancer cell is cancer and not normal is because of genetic alterations or mutations in its DNA sequence. And so what I've highlighted here is that when a change in the DNA sequence occurs, this is a DNA mutation. The consequence of that is that that change in the DNA base can actually lead to a mutated protein. So one change in the DNA can lead to actually a change in the protein sequence. If there's a change in the protein sequence, that actually makes it different. Now it's not a self protein, it's actually a foreign protein. If this mutant amino acid or this peptide is then presented on this HLA molecule, as I've previously shown, then again, there's a small population of T cells within a patient's body that has the right T cell receptor that can now bind to this mutation expressed by the cancer cell. So this is what we mean by, a T cell recognizing a mutation or a neoantigen. And we think, again, in these patients that had these durable, long-lasting responses, we could find T cells that targeted the patient's own unique DNA mutations. And so while melanoma is a deadly disease, the majority of cancer-related deaths are due to patients with metastatic epithelial-derived cancers. And so when I say epithelial-derived cancers, these are some of the common cancers that you've all heard of: colorectal cancer, breast cancer, pancreatic cancer, cholangiocarcinoma, et cetera. So the question at the time when I was training with Dr. Rosenberg was, well, can we use this knowledge of these mutation reactive T cells and harness them to treat patients with epithelial derived cancers? And so this really is where I'm going to describe Melinda's scientific story that has really ushered in our efforts to develop this therapy further. So Melinda is really the first patient with epithelial derived cancers to be treated with T cells targeting a unique mutation. And so many of you know her story. So she was diagnosed with cholangiocarcinoma that was metastatic to liver and lungs. And she had really kind of standard chemotherapy regimens: cisplatin and gemcitabine, Taxotere. For her, we actually generated, when she enrolled on the clinical trial at the NCI, we generated a TIL product. So we took her tumors, minced them up, and grew them up as I described in that cartoon I showed. And so we generated 42 billion TIL, and we infused those 42 billion TIL, followed by 4 doses of high-dose interleukin-2. We found out later that about 25% of this TIL product targeted a mutation in a gene or a protein called ERB2IP. And this is a mutation only found in her cancers and not found in any other person's cancer. So it's truly unique, personalized mutation. And so after infusion with these 42 billion TIL, this is our lung CT scan. You can see, again, metastatic deposits in the lung. And this is 7 months after TIL infusion. You can see that they've shrank, as shown here. In her liver, also evidence of tumor regression highlighted by these large yellow arrows. 7 months after therapy, you can see that these liver lesions have also shrank. And so after the infusion of what we call bulk TIL, this is what it kind of looks like graphically. So on the y-axis here, this is tumor burden, so essentially how big the tumors were. And then this is when the cell infusion happened, looking at the size of her lung, liver, and the total tumor burden. And you can see after cell infusion, 7 months later is when the scans happen. So I showed you the maximum tumor reduction. But you can see that her response lasted for about a year, 13 months of stable disease, what we call stable disease clinically. But you can see that the lung lesions started to grow after this 13-month time period. So we really asked ourselves, can we do better? And so based on what I've told you in terms of the potential importance of these mutation reactive T cells, this first infusion product had about 42 billion total T cells. About 25% of those were mutation reactive. So when I say can we do better, can we manufacture a product that's maybe a higher frequency of these mutation reactive cells and maybe a larger number? So her first infusion product was actually made up of 5 different, what we call TIL cultures, or these T cell cultures. So those fragments I told you, we put individually into wells of a plate, and we grew them separately. And her first infusion product, we actually pooled 5 of those wells, essentially. But it turned out that one of those cultures was highly enriched in the mutation reactive T cells. And so for her second treatment, we actually took that one culture and then exclusively expanded that one culture for treatment. And so after expansion, about 95% of the T cells within that infusion product were reactive against the ERB2IP mutation. And so her second treatment, we infused 126 billion T cells. This is a very large number of T cells. About 95%, or 120 billion, were reactive against the ERB2IP mutation. And so this is her lung CT scan. I'm showing you prior to her second infusion, October 2013, and actually 2 years later, a CT scan. And you can see that there is a large reduction in the tumor size, as shown by her lung disease here, and then afterwards, 2 years later. Many of the lesions shrank substantially, some disappeared. And if you look at it overall, she had actually a 35-month partial response. And that's shown here in this graph. So again, this graph shows a tumor burden on the y-axis, and then stratifying on lung and liver disease, the size of lung and liver disease in these different icons. And you can see prior to her second infusion, the lung lesions were kind of taking off, right? But then after infusion of these 120 billion ERB2IP mutation reactive T cells, you can see a dramatic precipitous drop in the size of her lung lesions. And you can see that the response lasted for about 35 months. So at that point in time, we saw clear evidence that these mutation reactive T cells could mediate tumor regression, but the reality was that the tumor started to slowly grow about 35 months later. And so we asked the question, well, why did these tumors progress? And can we give Melinda another effective treatment? And so what we did was at about 35 months after the second cell infusion, we actually resected some of Melinda's lung lesions. And so one of the lung lesions pathologically did not have tumor in it. So when a pathologist took a look at this lesion, there was no tumor. And so this was essentially a normal cell product. And so the first question we asked, well, is the mutation that's being targeted still being expressed by the cancer cell? Because one way a cancer cell could evade this response is that maybe it just lost the mutation, or a subclone of this tumor cell grew out and no longer has a mutation. So if we wanted to, for example, give another therapy targeting this mutation, we had to make sure that the mutation is expressed. And so we sequenced the tumors to see if the mutation is still there. In this normal cell product, there's only an A here, and this is a wild type sequence, so there's no mutation. So ATCG, these are the bases of DNA, and so we sequenced A is what's supposed to be there in a normal cell, and that's what's there. When we looked at the 3 lesions that were the 3 progressing lesions, we can see that there's actually both A and a G at both this location in the sequence set. So this signifies that there's actually a mutation in this ERB2IP gene. So the mutation is still present in the 3 tumor lesions that were progressing. So I guess that means that potentially if we wanted to retreat with the same cell product, then that could still be a viable option because the tumors are still expressing the mutation. So this is a bit of a busy slide, but I'll try to walk you through this. And so the next question we wanted to ask is, we infused a large number of these T cells in Melinda, right? Are the T cells still there, right? Because in theory, if we infuse them, they could either still be there or they could disappear. And if they disappear, then there would be rationale for infusing more cells for her next treatment. But if they're still there at high frequencies, then maybe that's not the limitation. That's not why the therapy is failing. And so this graph here, if this axis here is essentially just looking at the frequency of the mutation reactive T cell. So if you go up here, that means there's more of the mutation reactive T cell. And this is just a time or days after relative to the first cell transfer. So the bars, the gray bars, are essentially the infusion product. So the first infusion product is shown here. About 25% were mutation reactive. Here's a second infusion product, about 95% reactive. The red dots is the frequency of the mutation reactive T cell in the blood. So after infusion, the first infusion, you can see that there's the frequency of the reactive cells are about 5% or so in the blood 2 weeks later, and then over time kind of drops to about 0.01%. The first resection showed that the mutation reactive T cells were still there at between 1 to 10% frequency. After infusion with the second product, again with 120 billion TIL, you can see the persistence of those cells is much higher. So even a month later, in the blood, there's about 30% of the mutation reactive T cells. And if you follow it out all the way out to the second resection of her lung, so about 35 months later, you can see that in her blood, about 3% of all her circulating T cells in the blood are mutation reactive. And so that's actually a lot of cells, right? 3% of, again, the billions and billions of cells in her body are the mutation reactive, and it's ranked #1. So it's the most dominant T cell clone in Melinda's body. So it seems like the T cells are in her blood at very high levels. For the surgically resected sections, the ones that I showed you, the 3 lesions that we resected, you can see that the mutation reactive T cells are about 50% of all T cells within that tumor. So the T cells are at high levels in the blood, but also very high levels within the tumor. So it's not, so the reason that the tumors are progressing, but this is in spite of the fact that there's a lot of these mutation reactive T cells in the tumor and in the blood. So what's going on? So the next question we asked was, well, there's a large number of these mutation reactive T cells in there. Is there evidence that maybe they're being inhibited, right? Because tumors have multiple mechanisms to shut down T cells. And so with this, I want to highlight a very important pathway in T cells. And many of you probably heard of this pathway. It's called the PD-1 pathway. And so just highlighted in this cartoon, simple cartoon, again, here's our T cell. In addition to the T cell receptor that can recognize the cancer cell, there are many other receptors expressed by the tumor cells. And this is a receptor called PD-1. And upon engaging its co-receptor called PD-L1, what happens is a T cell then receives a negative signal. So it actually shuts down the T cell. And in doing so, that inhibits the ability of that T cell to kill or produce cytokine or function to mediate tumor clearance. And so with this pathway in mind, we asked whether or not, well, we've got a lot of these mutation reactive T cells in the tumor in the blood. Well, are they, expressing high levels of this PD-1 molecule, maybe getting shut down by this pathway? And so this is probably the most scientific type of graph I'm going to show you today, but this is what we call a flow cytometry plot. And so I'll explain what this actually means. And so these populations here, these different colors, they're actually analyzing single cells. And so what we did was for each tumor that was resected, we made a single cell suspension. So we actually minced it up and then digested the tumor, and then we extracted all the T cells from the tumor. And then we ran it through this technology called flow cytometry, which allows us to interrogate, okay, well, these cells, these T cells in the tumor, what are they expressing, right? And so on the x-axis here, we have, this is just called V beta 22, but this is jargon for essentially, this is our way to track the mutation reactive T cells. So anytime you see a shift on this axis, that means that's the mutation reactive T cell. So for example, these are the non-mutation reactive T cell. These are the mutation reactive T cells because they express V beta 22. On this plot here, we have PD-1. So anytime you see higher levels of PD-1, you'll see a higher expression on the y-axis here. And so as you can appreciate from the tumors 1 and tumors 2 and 3 and the blood, when we look at PD-1 expression on the mutation reactive T cells, you can see that compared to the non-mutation reactive T cells, there's actually elevated amounts of PD-1, especially in the tumor. You can see here, these are the mutation reactive T cells. They express much greater levels of PD-1 compared to the non-mutation reactive T cells here, right? So hopefully you can appreciate that this population is kind of shifted higher up on this axis. So this means that indeed the mutation reactive T cells are expressing PD-1, high levels of PD-1. But according to that cartoon I showed, PD-1 is one component, but the other component is PD-L1, it's co-receptor. So there has to be an engagement of PD-L1 in order to cause a shutdown signal in the T cell. So what we did was we resorted to something called immunohistochemistry. And what this is actually a process where the tumor is embedded in formalin, and then it's sectioned into very thin sections and put onto a glass slide. And then on that glass slide, we can interrogate expression of certain proteins. And we can do this for PD-L1, for example. And so that's what we did. And so what you can see here is anytime you see a brown staining, that's indicative of PD-L1 expression. So that's clearly you can see a lot of brown here. When we do high magnification, you can actually see cells that have membrane expression of PD-L1 within the tumor, right? And so we can conclude that there's strong PD-L1 expression. And when a pathologist looks at it, they can actually tell us that it's actually on macrophages. It's another type of immune cell that actually infiltrates tumors. PD-L1 expression is on the macrophages, not really on the tumor cells. But what this means is that the T cells engaging macrophages, there could be a PD-L1, PD-1 interaction and potentially shutting down the T cell response. So at this point, we had all this data, right? And we were trying to formulate, okay, well, should we treat Melinda with more cells, another adoptive cell transfer, or maybe are there alternative options, right? So, to summarize at this point, we found that, Melinda had still high levels of mutation reactive T cells. A lot of them are in the tumor, 50% of all the T cells in the tumor. In the blood, lots of these mutation reactive T cells, and they all express PD-1. We know that the mutation is still there in the tumor, and we know that the macrophages are heavily infiltrated in the tumors, and they express high levels of PD-L1. So as many of you know, there's actually another immunotherapy drug that can block PD-1. There's actually multiple drugs that can block PD-1. And so we thought instead of giving Melinda more cells, because it looks like she had a lot of cells already in her body, we decided to treat with anti-PD-1 therapy. And so upon anti-PD-1 therapy, Melinda received six doses total. Here's a lung CT scan. And so when we track these two lesions here, this is 3 years out, you can see that this lesion had disappeared. This lesion actually disappeared relatively quickly. This lesion here has remained the same for the last probably 5+ years. And so she had complete regression of one lesion, and another lesion shrank and has remained stable for over 6 years after the last anti-PD-1 dose. And we hypothesize that this is likely scar tissue that just never resolved. And so, so that's really, Melinda's scientific story in terms of what we did in terms of looking at mutation reactive T cells, transferring those T cells, understanding the biology of why maybe it didn't work for a durable, complete response. And that led us to treatment with anti-PD-1, which, thankfully has led to a clinical response for Melinda. So Melinda was the first case, right? And we were certainly, as scientists and researchers, very excited for the promise of targeting mutations with T cells. And we had success in other patients as well, a handful of patients. And so here's just a brief summary, right? So I've outlined Melinda's case, which is shown here. These are patients that have responded to neoantigen-directed TIL therapy. Another patient with colorectal cancer, we were able to generate TIL that targeted oncogenic mutation in KRAS G12D, and she actually had a response as well. She had a 9-month partial response. We resected the one lesion, and to this date, to my knowledge, as of at least last summer, she was still no evidence of disease. Colleagues of mine treated a patient with cervical cancer, breast cancer with neoantigen reactive TIL, and they experienced complete response. And so, these are the patients that responded. the harsh reality is that, not all patients respond. Many patients we treated with this sort of strategy. There are nuances, obviously, but many patients treated with this sort of strategy did not respond. So we do have work to do. And so there are challenges with TIL therapy. So what I've outlined to you, as you might appreciate, is highly personalized, right? And somewhat complex. It's not taking a drug off the shelf. You have to get tumor from patients, we have to grow them out. There's long manufacture time. And so there are challenges. And so the challenges with TIL therapy, as I just mentioned, it's not effective for many patients with metastatic epithelial cancer. And this could be due to a variety of reasons. One could be the frequency of the tumor reactive TIL could be highly variable. In some cases, after growing out the TIL, only a very small percentage of those T cells can recognize cancer. Another variable is that the targets, the antigens that we target might be only expressed by a fraction of the cancer cells. And this is kind of highlighted by this diagram here called tumor heterogeneity. So tumors are actually very complex. And so a tumor cell can be heterogeneous, or tumors can be heterogeneous, meaning that a tumor mass can be made of different tumor cells with different genetic makeups. And so just for simplicity's sake, if we're targeting tumor cells that express the target only in the yellow cells here, then the blue and the red ones will not be targeted, right? So that's the concept of tumor heterogeneity. Not all tumor cells are the same. And in the setting of T cells or TIL therapy, the phenotype of the TIL, the phenotype essentially the T cell age and how they look can be quite different. And in the setting of TIL, they could be what we call more differentiated or older T cells because they've been actually in the tumor for a long time fighting that battle. But for some reason, they could be there for months or maybe even years. And in this hostile environment, fighting the battle, losing, getting exhausted and getting older. And so once we take those out and grow them out, they might not be the best cells to give back to the patients. And in preclinical models, this is really shown here. These are just different T cell subsets. And on this spectrum here, these are kind of older cells and these are younger cells. In preclinical models, when you actually use the younger cells for adoptive transfer experiments, these younger cells can clear tumors much more effective than if you transfer these older cells. And so again, in TIL therapy, most of the cells are probably closer to this spectrum here rather than this spectrum. The functional capacity of the tumor-reactive receptors can be highly variable. Again, we've got billions of T cells and each one has a different receptor. That receptor can have different functional abilities. And so it can be highly variable in TIL. And of course, you need surgical resection in order to get the tumor and the TIL out. So how might we overcome this sort of limitations of TIL therapy? So one major strategy that we're pursuing here in my lab is something called T cell receptor gene therapy. And it's actually a relatively simple concept, I think, to grasp. So I mentioned that T cell receptor is really one of the defining characteristics of a T cell because it can tell you what it recognizes, right? That T cell receptor tells what that T cell, what to target. And so, for example, what we can do is take peripheral blood from a patient. And what we can do is take a T cell receptor that can target a known tumor antigen and then genetically insert it into the peripheral blood T cells of that patient. In that way, we're redirecting all those T cells from the blood to now target the tumor cell. And then we expand all those T cells up, as I've kind of described, and reinfuse those back into the patient. And so with this sort of therapy, no surgery is required. We can control the phenotype, the age of the cells a little bit better, because within the blood of everyone, we've got, younger T cells that are not exhausted, haven't been fighting, the cancer cells. And so we can insert this T cell receptor into these hopefully more potent T cells. We can generate a high frequency of these tumor reactive T cells because with our current technology, we can get this receptor in to T cells at, about 70% to even 90% efficiency. So we can essentially turn about 70 to 90% of all those T cells in the blood into tumor fighters. And we can use well-characterized T cell receptors that can target things like driver mutations, right? Many of you probably heard of some driver mutations. These are mutations that are critically important for cancer cells. And so if they're very important for the cancer cell, they're less likely to lose that, that expression. And so there are driver mutations in things like KRAS and P53. These are maybe some molecules that you may have heard of. There is a major challenge, and that's really that this technology is currently limited to targeting tumors that express that specific mutation and HLA molecule. So this MHC molecule here, HLA and MHC are interchangeable, but I'll just spend like 2 seconds to explain this molecule. Within the human population, there are thousands of different MHC molecules, right? And so a given T cell receptor only can recognize, in theory, one MHC molecule and that mutation. So this really dwindles down the eligibility pool for a given T cell receptor, right? Whereas TIL, it's personalized for that patient. That's wonderful. But for this approach, that patient's tumor has to have the right mutation and has to have the right HLA. And so again, it's a very small piece of the pie at the end of the day. And so for the remaining little bit of time, I just want to briefly highlight, what we're doing with TCR gene therapy here in Portland. And so I want to highlight a report that we put out last year in the New England Journal of Medicine, where we demonstrated proof of principle that this might be a feasible path forward. And, the patients we treated were pancreatic cancer patients. We highlighted one patient here. There were 2 patients we actually reported in this brief report. And so the patient that we highlighted in this report, 71-year-old female diagnosed with pancreatic ductal adenocarcinoma, so the most common form of pancreatic cancer. Many of you know that this is a very lethal disease. She underwent very standard treatments, Whipple resection with chemotherapy. She underwent TIL therapy, clinical trial, but a growth of tumors were observed 6 months later. We were able to genotype, do molecular studies, and find out that her tumor was KRAS G12D positive. G12D is a mutation. And HLA typing revealed that she was HLA C0802. And I bring these points up because in a previous report, a previous patient that we treated at the NCI, we found T cell receptors that targeted KRAS G12D in the context of HLA C0802. And so what we did was we wrote a single patient IND to treat this patient with those receptors that we found in another patient, that other patient with colorectal cancer. I'm not going to go into details here, but I just want to highlight that this is how we generate the cells. We took her peripheral blood, then we engineered them. So we use a virus to insert the T cell receptor. And then we grow those cells out to about 10 days. When the patient's ready, we thaw those cells out, and then we grow them for another 14 days, 2 weeks, to generate billions of T cells. In terms of the treatment scheme, we preconditioned the patient with cyclophosphamide, which is a chemotherapy regimen. And then on day 0, we infused 16 billion T cells, and about over 90% of those T cells expressed a TCR that targeted mutant KRAS. And then we gave her 5 doses of high-dose interleukin-2 afterwards. And so after that treatment, you can see this is our CT scan of her chest, showing you 3 lesions here prior to treatment, and then on 3 months and 6 months later, you can see that the lesions have shrank substantially. And so she has an overall 72% reduction by RECIST, which is a way we measure tumors, at 6 months post-cell infusion. When we look at persistence of the engineered cells, so this is, again, here's just level of persistence on the y-axis, and then time after cell transfer. We can see that at close to 6 months after cell transfer, about 2% of all her circulating T cells were the engineered T cells. And so just to update for this patient. So this patient experienced a 1-year response to her therapy, but then she did have slow progression in a few of her lung lesions. So we decided to retreat her with more cells. We made a few modifications. One was that we added anti-PD-1 therapy, hopefully to boost the T cells once they're transferred. And we actually used 1 TCR instead of 2. So I forgot to mention that the first therapy, we used 2 different T cell receptors. We chose to do 1 the second time because in our lab work, we found that this 1 TCR might be more functional than the other T cell receptor. So those were the 2 changes we did. So this is a scan just 1 month later. So 1 month after cell infusion, we do see evidence of shrinkage. You can see our tumors are shrinking here. the tumors have shrank here as well. When we look at persistence of her engineered cells, you can see that after her first infusion in black, spiking at about 2 weeks and then decayed over time. After her second infusion, her peak level of transduced or engraft T cells was actually lower. And at 3 months, I can tell you that it's slightly lower, maybe about 1%. So we're still trying to understand that a little bit better. We think we gave her a more potent product, but it looks like in terms of persistence after transfer, after her second therapy, it looks like a little bit less. But all in all, the patient currently has stable disease at 3 months, 25% reduction in RECIST after her second infusion. And so we're kind of eagerly awaiting her 6-month scans, which is coming up very soon, and hope and knock on wood that she continues to respond. If not, we're going to obviously try to understand her tumor biology better, and hopefully we can manufacture another therapy for this patient as well. And so with that, I'd like to conclude that the adoptive transfer of TIL, or TCR engineered T cells targeting mutations, can mediate regression of metastatic epithelial cancer, including cholangiocarcinoma. I do want to stress, though, adoptive cell therapy targeting mutations has been ineffective for many patients, and thus improvements to adoptive cell therapy are needed. Factors such as tumor heterogeneity, as I touched upon, which means variable expression of the target, the T cell phenotype, how they look, the functional capacity and persistence of the T cells, and the hostile tumor microenvironment can impact the efficacy of adoptive cell therapy. And so this is something that we are quite interested in, we and others, obviously, is that you can manufacture cells, manufacture potent cells, but once you put them back into the patient, each patient has a different microenvironment that the T cells are going back into. This is a busy slide, but I just want to highlight that tumors are a complex mass of cells and factors. And so in this diagram, the purple are the tumor cells, but there are other types of cells. MDSCs, or myeloid-derived suppressor cells. And so, as the name suggests, they can suppress immune responses. Regulatory T cells in blue here can also shut down responses. Macrophages that I've alluded to. In that stain I showed you, lots of macrophages are in the tumor. And we know that macrophages within tumors can actually be bad players. They can shut down T cell responses. And then these are different cell types. And then there's soluble factors that are produced by some of these cells that shut down productive anti-tumor responses. And so this is kind of the next frontier, is how do we engage or overcome some of these immunosuppressive factors once the T cells are transferred. So some of the work that my lab is doing is trying to characterize the immune response in patients that respond or don't respond to adoptive cell therapy, so that we can identify these mechanisms of why therapies fail some patients. We're also trying to find additional T cell receptors that can target other tumor-specific antigens. So I mentioned that each T cell receptor can only be used for a certain population of patients based on their mutation and HLA. So if we can find additional T cell receptors, maybe we can build a library, as we call it, of T cell receptors to treat more patients. We are trying to, for lack of a better term, armor these T cells so that they can function better in the tumor. So we are devising synthetic receptors that we can engineer into T cells that we hopefully can enhance their function once they're in this hostile environment. And we're trying to develop ways to grow T cells better. So we're, depending on what factors you put into the culture, you can actually make different T cells of different functional ability. And to the last point that I mentioned about the tumor microenvironment, we're trying to find ways to enhance cell therapy against cancer through targeting the tumor microenvironment. And our current clinical trial is using these engineered T cells to target hotspot mutations. And we're combining it with something called anti-CD40 and anti-PD-1, because anti-CD40 has been shown to be able to activate some of these macrophages or dendritic cells, or these antigen-presenting cells. So we're hoping that by re-engaging those immune components in combination with cell therapy, that will tip the balance in favor of tumor rejection rather than tumor progression. So with that, I'd like to conclude my talk. Again, thank you for the kind invitation to be here, and I'd be happy to answer any questions.
That was a great explanation. I always learn so much when you give these presentations. We do have quite a few questions, so I'm just going to start at the beginning. What mutations or neoantigens that these T cells recognize have been identified? I think you covered that.
Yeah, I can expand on that though.
Okay.
So there's two approaches to targeting mutations or neoantigens. One is a highly personalized approach, like what we did for Melinda, right? So what we do is we take a tumor, we can sequence the tumor, identify all the mutations expressed by the cancer, and then try to find T cells that target those unique mutations. Most of those mutations, of course, is only in that patient's cancer and not expressed by other patients' cancer. So highly personalized approach. So in that sort of, to answer that aspect, we can usually find T cells that target a patient's own unique mutations. And if you take the work of the surgery branch, the NCI, and others around the lab, we found hundreds and hundreds of T cell receptors targeting, different mutations. From the shared mutation perspective, so these hotspot mutations like KRAS, P53, where there are recurrent mutations at the same location of that gene every single time, we are starting to build a library now of T cell receptors that can target KRAS G12D, KRAS G12V, P53, R175H, these hotspot mutations. And so that library is growing. I can't tell you the exact library and the coverage of mutations in HLA, because HLA is the other aspect, right? So you have to have the mutation, but then HLA, and then a given T cell receptor can only recognize that mutation HLA. But what I can say again is that we do have receptors now that can target some of these hotspot mutations in relatively common alleles, so that at least some patients will be eligible.
Thank you. Does the gut microbiome influence response to adoptive therapy the way it does in immune checkpoint inhibitors?
That's an excellent question. I think maybe just a brief background on that. I think just relatively recently, the microbiome, essentially, the bacterial component of our bodies, right, have been shown there seems to be quite strong correlative evidence and some preclinical data that suggests that it does impact, things like immune checkpoint blockade, anti-PD-1 therapy, anti-CTLA-4 therapy. That's an area of investigation that's, I think, very interesting and exciting, worth pursuing in something like adoptive cell therapy. In the setting of human adoptive cell therapy, I've not seen any data that is looking specifically at whether or not the gut microbiome of patients is impacting efficacy. I think studies in the future should be designed so that we can be able to obtain those samples from patients that are undergoing cell therapy to see if there's any correlation. The major stumbling point for adoptive cell therapy is that these therapies are, it's hard to treat a lot of patients like you can with immune checkpoint inhibitors. So these studies will take a long time and require probably a lot of patients in order to get some sort of evidence that gut microbiome might be or might not be playing a role in adoptive cell therapy.
Okay. Is there any way to test to see if there will be a response in a particular patient prior to infusion?
I wish we had that answer. And it's really, are there predictive biomarkers? The question is, are there predictive biomarkers to stratify patients that are more likely to respond? And so in the setting of immune checkpoint inhibitors, there are some biomarkers that, might stratify patients that are more likely to respond. And so things like the mutation burden, right? So if a patient's tumor has a high mutation burden, they are more likely to respond to some immune checkpoint inhibitors. For adoptive cell therapy, I think we're in the same boat as, the microbiome, where we just haven't been able to treat that many patients to find those types of biomarkers. There are some, I think, very basic things that we can look at, right? So the way a T cell can recognize cancer cells, we know some of the basic mechanisms, right? The TCR needs to see the MHC molecule, has to see the peptide, the mutation. So those are things that we can evaluate, for example. So if a patient comes in, potentially eligible for a given T cell therapy for KRAS, for example, we can check the tumor to see if it's expressing HLA or that MHC molecule and the mutation. And if it's not being expressed, then I think that's a clear exclusion criteria, because that T cell receptor is not going to work for that patient that doesn't have that right. If the tumor has lost that MHC, for example, or lost that mutation, then that is not likely to be effective. There are other, without going into more details, but there are pathways within tumor cells that can be mutated that could probably make it less likely to respond to adoptive cell therapy. But there have not been good studies to prospectively kind of identify these biomarkers.
Okay. Do you combine ACT with cytotoxic chemo to generate or create an immunogenic cell death?
Yeah, that's another good question. I think a lot of these questions are very, there are people that have been reading up on literature. So I think, so immunogenic cell death. So in our, historically, in adoptive cell therapy, we preconditioned patients with cyclophosphamide and fludarabine. Some of the major reasons for that is to temporarily wipe out the patient's immune system so that when the cells are transferred, they'll have the best transfer engraftment because there's less competition, right? So they get in there, there's more cytokines, more proteins that can encourage engraftment of those T cells. It turns out that cyclophosphamide is also a potential immunogenic cell death chemotherapy, right? So there could be, cyclophosphamide could also be potentially impinging on that pathway to help adoptive cell therapy. There are a number of different immunogenic cell death immunogenic cell death chemotherapy, some platinum-based chemotherapies preclinically that have kind of looked promising in terms of making, causing cells to die, tumor cells to die in a way that, flags the immune system to recognize it and kill it. We have not extensively looked at that component, right? So the lymphodepletion regimen has been the most tried and true. It would be wonderful if more groups were able to test different chemotherapy regimens for either lymphodepletion and or immunogenic cell death to hopefully help the cells once they're transferred. We've had a couple experiences using like gemcitabine with cyclophosphamide. In a way, gemcitabine has been also. There's evidence that gemcitabine can cause a immunogenic cell death, and the patients did not respond, but it's, you can't really make too many conclusions from one or two patients, especially when they're late stage pancreatic patients, with poor performance status. So I think that's a very interesting question to explore, is to try to figure out what's the optimal chemotherapy, potential immunogenic cell death regimen to combine with adoptive cell therapy. I think preclinically it can be done relatively easily, but in humans it's a little bit more difficult.
Yeah. Is the TCR trial at Providence similar to the one at NCI?
Yes, it is. So it's similar in the sense that we're using similar T cell receptors. So targeting mutant KRAS, P50. NCI does have a P53 TCR. There is one major difference though, is that the NCI is still using the lymphodepletion regimen of cyclophosphamide fludarabine. Our current trial is not. And so we're testing whether CD40, engaging that CD40 pathway, activating these dendritic cells, macrophages, et cetera, can either replace or be at least a substitute for the lymphodepletion regimen. So that would be the major, I would say, the treatment difference. But in terms of targeting, we are using similar receptors. The NCI trial also, I believe, has a personalized TCR trial where they actually will try to find TCRs unique to each tumor. So that's something that we're not currently really set up to do well here, based mainly on bandwidth. NCI has a little bit more capacity than my lab here in Portland.
Okay. Are only those invited into a clinical trial eligible for this treatment? And what are the qualifications, to your knowledge, what cancer centers are sponsoring this type of treatment?
So, so with TCR gene therapy, the critical molecular eligibility will be expression of the mutation. So the cancer must express the mutation targeted. and the HLA type of that patient. So again, with TCR gene therapy, those are two molecular eligibility criteria. And then on top of that, there's going to be eligibility criteria based on, just clinical performance status. But when you look, that's for TCR gene therapy, and that's universal for any T cell receptor gene therapy type of trial that's targeting mutations. But there are TIL therapy trials, and in terms of TIL therapy, there won't be that restriction, mainly because that therapy is based on the patient's own T cells and the patient's own T cells and their own receptors, and so there isn't any requirement there. In terms of for clinical trials in general, typically the process is to identify a promising clinical trial, and clinicaltrials.gov is a pretty good resource for that. And then it's really just reaching out, either yourself or your oncologist, to the study coordinator to see what are the eligibility requirements. And so and then that initiates a process of potentially going into a clinical trial. The invitation thing, I'm just usually most clinical trials are based on the fact that you or the oncologist will engage with the clinical trials team for that particular trial, and usually it's not an invitation, to my knowledge.
Okay. What are the risks to the patients with using TIL, side effects, adverse events?
Yeah, so interestingly, the majority of the toxicities of these TIL therapies, the classic TIL therapy, is not really to the T cells themselves. The T cells are relatively innocuous in terms of side effect profile. It's actually the lymphodepletion regimen, so the preconditioning regimen that I was referring to, the cyclophosphamide fludarabine. These toxicities are hematological in nature, so blood toxicities, because it's just temporarily wiping out your immune system. So all your blood cells are going to be wiped out temporarily. These are all fully reversible, typically in most patients. And so, but it is, that would be the major side effects. In trials that include interleukin-2 therapy, as Melinda's trial, when she was treated, included, there are toxicities mediated by IL-2. And those include things like hypotension, vascular leak syndrome, extreme fatigue, hallucinations, and some other side effects as well. But these typically, again, are fully reversible within a few days' time. But again, in terms of toxicities, the T cells themselves usually do not cause too much toxicity.
Okay. Would you please explain the difference and compare TIL therapy with CAR T therapy?
Yeah, absolutely. So TIL therapy, as outlined in this presentation, is taking out a tumor, throwing out those T cells from the tumor, and then reinfusing those back into the patient. So there's no gene modification, right? So it's just your endogenous T cells, hopefully growing out the tumor reactive ones and giving them back. CAR T cell therapy, CAR stands for chimeric antigen receptor. And this type of therapy relies on gene engineering of T cells to express a CAR, chimeric antigen receptor. This receptor is, for lack of a better term, a Frankenstein protein. That's why it's called the chimera. And so on the, if you can imagine a receptor, the receptor part that engages the target is typically based on an antibody. And antibodies can bind to cell surface proteins, for example. That portion is then stitched onto a transmembrane domain, which allows it to insert into the cell membrane. And then the intracellular domain can be variable, but typically they are essentially T cell signaling domains. And so what you have in this chimera or Frankenstein protein is a receptor that can engage something on the cell surface, and once upon engaging, can send positive T cell activation signals to the T cell. And so I would say one of the major differences is that the CARs can recognize cell surface proteins directly, right? So the T cell receptor, or TILs, recognize the mutation or the peptide when it's presented by that MHC or HLA molecule. So that means that those TILs, or those TCR engineered cells, are HLA restricted. They need that HLA. The CARs do not. So they just bind to some cell surface protein. CARs for CARs for blood cancers have been very effective, right? CARs for solid cancers have so far been quite ineffective, actually. And one of the reasons why we think that is that when you think of a cell surface on the cancer cell, there are very few cell surface proteins on a cancer cell that are not expressed by some normal tissue in your body. And so finding something to target, it has been very difficult for chimeric antigen receptors or CAR T cells, because if it's expressed by your normal tissues, that T cell will also attack your normal tissues and cause toxicities. What you want is to attack something on the cell surface that's only expressed by the tumor cell. And again, that's been very difficult to find. Something exclusively expressed on the cell surface of a tumor and not on a normal cell.
Okay, a couple more questions and then we're going to let you go. Is there an age cutoff for the trial?
For our trial, that's a good question. I think it's, I should know this, but our clinical lead actually wrote the clinical component of this. I believe it's 70 years old. Okay. But I would have to confirm with that. But
okay, we can look that up on.
Yeah, we can look that up. Apologize, but again, my Dr. my clinical colleague wrote that.
Okay, and then there's a question that relates to having resected tumor already. Could TIL therapy be created from a tumor that was already resected?
Unfortunately, unlikely, mainly because once it's resected, it's either going to go into formalin, which is a fixative, or it's going to be frozen away somewhere in a freezer. So once that tumor has been processed that way, it's hard to grow out the viable T cells that are required for the TIL therapy. The next generation approach is that potentially what you can do is identify the receptors. So, from the tumors, even if it's fixed or frozen, you can sequence or find the T cell receptors and then reconstruct those T cell receptors and put it into peripheral blood T cells. So that's the next generation, which we are not quite there yet, but there is a company in Europe that's evaluating that sort of strategy. But it's it's certainly more difficult than the standard TIL therapy.
Okay, I swear, last question. Would you please discuss TCR therapy against mesothelin and whether this might be an effective target against cholangiocarcinoma?
Yeah, that's a very good question. I think so. As alluded to with the chimeric antigen receptors, right? So with chimeric antigen receptors, they've been limited to cell surface antigens that are typically overexpressed by tumor cells. And one of these proteins happens to be mesothelin, right? So chimeric antigen receptors have been tested to target mesothelin in clinical trials. And so far it's been, again, relatively ineffective, unfortunately. Could a TCR be more potent against mesothelin? Potentially, yes. So maybe if you found a potent T cell receptor, which I believe a group in Seattle has found, maybe this T cell receptor could more potently recognize the mesothelin expressing cell. My major concern is that, in the CAR studies, chimeric antigen receptor studies, there was very little hints of clinical activity, and there was very little hints of toxicity. But my concern is that mesothelin is a normal protein that's expressed by some normal cells. I think the pleura, the cardiac pleura, and also the lung pleura, I believe. So my concern would be if you can get potent targeting of mesothelin, you might also get what we call off tumor on target toxicity, which means that yes, it might hit the tumor, but it might also hit the normal cells. And so that would be my major concern with that sort of approach, because mesothelin, again, is expressed by normal tissues.
Okay. Thank you, Eric. We appreciate this presentation. We've learned so much. If any other questions come up, I might send them to you for some answers. Yep. And again, I am indebted to you for my life, and I'm so grateful for you, and I appreciate the time you took for all of our patients today.
It's absolutely my pleasure, Melinda, and I'd be happy to answer any questions. Again, Melinda, send them my way. I'm happy to help any way I can.
Perfect. Thank you. Everyone have a wonderful day. Thank you.
Okay, bye-bye.
Bye-bye.